Okay, thanks so much, Keir. It's really wonderful to be here. And just thanks to Joe for kind of opening up the meeting this morning and pointing out some of the headwinds that I think we're all feeling. And I think in this moment in particular, it's just great to be together. The turnout here, spectacular. I'm finding inspiration in every conversation, every interaction. Let's just keep doing more of this. All right, so I'm going to try to thread two needles in this talk. The first is similar to what Jesse Bloom attempted to do, which is to give a talk that you've never given before, which is sort of a harrowing experience. And then the second needle here is that I'm going to bring in we're in Keir Bala country, the fellow who's just introduced this session. And so I'm going to bring in some of his work. And the thing about Keir is he's such a classy, killer, creative, just eccentric, bespoke craftsperson of a scientist that it really raises the stakes when you try to give a talk like this. So usually I can just fall back on my false professorial beard, but with Keir, he outflanks you there. You just at every turn, he's got you. So we'll see what happens. We're going to be talking about viruses swimming with the ciliates. I'm not going to be talking about flaviviruses that are cartooned in the corner there, but I can, growing off of John Scoggins' great talk, report that there's life after the PhD. So Ian Boyes, this fellow that discovered RTP4, just discovered from iguana genomes another restriction factor that blocks flaviviruses, E3 ubiquitin ligase that has an assist from a family of transposable elements. It's on the bioRxiv. Look up iguana and Boyes, and you won't be disappointed in that work. OK, so before we get to the viruses and the ciliates, I want to acknowledge some human beings. So a lot of the work I'm going to be telling you about today started with Katie Dietz, who's shown in the upper left corner there. Katie is a recovering mouse immunologist. She got her PhD in Russell Vance's lab and has picked up the ciliate Tetrahymena thermophila that I'll be telling you about today. Katie has been running this with a team of great undergrads, Richard, Coots, and Eleanor, some assists from Hannah Young, a great grad student in the lab. Drew Janik runs aquariums in the Elde lab. A couple of faculty who've been along for the science adventures here, James Gagnon, Jamie, who is doing a sabbatical in my lab right now. Kristen Kwan, who's really taught us about zebrafish, which will come into the picture as well, which means I can't avoid this fellow, Keir Bala, who's really the founder of all of this work and an impresario, I think, for sort of building these bridges in this science. Am I laying it on thick enough here, Keir? All right. OK, I got it. OK. And then a couple other postdocs, Stephen. Not yet. Well, yeah. Whatever heat comes from this talk, put it on me, not on Keir's shoulders here. OK. OK. So I'm not talking about flaviviruses, but I do want to talk about virus transmission, which is a completely new topic that I'm sort of jumping into a new space here. And thinking about the varied routes of virus transmission, all the way on the left here to direct contact, or so the notion that virus particles don't really need much help to sustain transmission chains of infection, all the way to really complicated replication styles involving Slavic and urban sort of settings, as we heard from Jesse earlier, with mosquitoes potentially in the mix. So sort of the big idea of this talk is that this is a continuum from sort of direct contact to like full-on vector, really complicated replication styles, but that there's probably a lot of virus transmission biology somewhere across this continuum, somewhere in the middle. And so I'm going to give two examples of this using chum salmon reovirus. This is a distant cousin of rotaviruses that shows up in the epidemics and cruise ships and things like this, causes diarrheal disease. And then another virus that's familiar in this territory, zebrafish picornavirus, that was discovered by Keir. The real star of the show is going to be Tetrahymena thermophila. So this is the model genetic ciliate, probably most famous for some of its genomic gymnastics. It has a germline micronucleus that develops into a somatic macronucleus. The chromosomes get shattered into pieces and amplified up. Blackburn and Greider got a Nobel Prize for purifying telomerase to solve the end chromosome erosion situation with Tetrahymena. It's also a master cell biologist. So echoing again on what Joe was mentioning this morning, The viruses are teaching us our own cell biology. These ciliates are also teaching us cell biology. It's sort of the fleas of the fleas coming together, I think, to teach the lessons of how this more complex systems virology might unfold. Okay. So let's talk a little bit about protists. And I should say, so the opening sort of attempt we've made here with Tetrahymena is to ask the question, could it serve in aquatic environments as sort of a maritime mosquito? Could it be vectoring viruses, or is it doing something kind of in between? So a little more introduction as protists as purveyors of particles. And the idea here is that these are really master cell biologists. At least two routes of inward traffic. One is phagocytosis at the oral apparatus that's shown up there. This is like concentrating the bulk of aquatic environments into phagosomes that then get processed as they go. There's also clathrin-mediated endocytosis arranged across the ciliary bodies. It's a really beautiful cellular architecture in at least three ways out: constitutive secretion, regulated exocytosis, which seems to have evolved in a suite of host or predator-prey interactions, which I don't have time to talk about today, and then also excretion, both of water through the contractile vacuole, as well as nutritional waste or nutritional particles through the cytoproct. So single cell, entire creature that's conducting all of this cell biology, purveying particles. We've talked a little bit about the nuclei or the genomic gymnastics, but it's really in the endomembrane trafficking systems where these things really sing out for the specialized biology. Let me put my money where my mouth is. So this is, I'm going to show a video in a second here of Tetrahymena just sort of circling around doing constitutive secretion. This is using a marker called escargot, which allows an ER bulk of protein to build up. You treat it with this ligand called SLF, and a wave of constitutive secretion goes across the cell. Tool is developed in Ben Glick's lab at Chicago, and then picked up by Aaron Turkowitz, my old PhD advisor, who just retired from the University of Chicago, who put this to express this in the ciliate Tetrahymena. Okay, so this is sped up about 3 times. Escargot is in magenta here. And what you can see is as the ciliate starts to circle, there's some remnants of that protein that's coming out of it and showing up as this sort of, I'm showing my age here, so I think of Charlie Brown Pigpen, who's like constantly covered in dust and sort of, and so when we just look at this under the light microscope, now you can start to see that escargot kind of shimmering behind the ciliate as it's making these circles here. You know, we look at these under light microscopes, it looks like these cells are just swimming around. In fact, they're both imbibing all of the particles in bulk around them, and then also conveying them back to the environment. They're purveying these particles. I hinted at some of the really cool organization of the ciliates. So their cellular architecture is arranged around the ciliary rows. This is because they're called alveolates. So they have an extra layer of membrane right underneath the cell surface that constricts where all of the traffic inward and outward can occur. So now we're looking again from the Turkowitz lab. Cells, transgenic tetrahymena, are expressing GEF1P. This is a guanine nucleotide exchange factor. And by the way, in all of the talks so far, we get these sort of alphabet soup or these laundry lists of all these ISGs, interferon-stimulated genes. Many of them catch the cell biologist's eye because these are large GTPases, large GTP proteins, proteins involved in all of this membrane trafficking. And so our immune system is trying to tell us something. It's written in our own cell biology as well. And I think you can see this sort of at the single cell level here in Tetrahymena. Okay, so GEF1P, this is a Golgi marker. I've started the video, and you can see it sort of perking along. There's little parts of the Golgi moving between. All of these dots are near the basal body, the base of the cilia. And all of the constitutive secretion, the regulated secretion is happening there, as well as clathrin-mediated endocytosis. We sort of go to the middle of the cell, into the depth of it, and you just get a sense of all of that sort of activity that's percolating just beneath the surface. Okay, so that sort of sets the table a little bit for the cell biology here, but now let's add viruses and start to think about what might happen. So some of the early work here is really focused on the notion that as these ciliates are concentrating particles from their environment, they're going to pick up viruses And the phagosome, there's all kinds of proteases and other processing going on here. And so this most likely means that as you purposely feed them or expose them to these viruses, that they'll be degraded. And that indeed seems like the case for this collection of phages, with the exception of Q beta, which, so we're going on the y-axis here. As you go lower, there's more degradation of the virus particles. That's not the case for Q beta, the phage, but for both the non-enveloped and enveloped mammalian viruses, it looks like it's clearing this. It might be consuming them or gaining energy from these virus particles. This has been proposed as ways of sort of bioremediating virus-infected water streams, and there might be something to that, given that. There also turns out there's sort of an interesting, and maybe not surprisingly, the idea that viruses have sort of flipped the script or turned the tables on this protease activity, in some cases to maybe take advantage of it, to start to process the virus forms in ways that could actually activate them. And so there's some really interesting evidence here with chum salmon virus, this reovirus, distant cousin of rotaviruses, where if you incubate them with ciliates, with or without ciliates, you see a different outcome in terms of the titer of the virus on the y-axis. And so cells or virus particles that are not pre-incubated with ciliates, you see sort of the same titer across the board here. Whereas given some time associated with a ciliate, it looks like the titers go up. Sort of a mysterious outcome here. The mechanism of this isn't described in the work so far, but sort of some of the directions this is pointing to is the idea that that virus is getting processed as it goes through the phagolysosomal system, and that then it comes out the other side, or as hosts in some cases are consuming the ciliates themselves as food, that there could be a route here. These virus infections were done in vitro, so the virus was given to the ciliate and then returned to a cellular infection model, and that's where you see the increase here. We actually couldn't believe it. Katie in the lab couldn't believe it, and so she set out to try this also with Tetrahymena thermophila in particular. I want to pause here just to note that the kind of species matters. So one of the reasons that Katie is really focused on this species in particular is because of all of the molecular genetic tools that are available in it. This is from the sort of Blackburn Grider days forward. You have the ability to do genetics, to manipulate the cell in ways that might reveal some of the mechanisms or bring out some of the virus biology here, the infection biology. Indeed, we were able to replicate this finding where the virus, chum salmon virus, becomes quote unquote activated after it goes through an association with Tetrahymena. Okay, so maybe some interesting clues that sort of virology could go in different directions as you sort of step between those models from all the way from direct contact to a vector scenario. But what's really going on here in the middle, and can we actually sort of deploy this in whole animal systems where we're putting together the real biology and not just sort of taking things potentially out of context? So enter Kiribala in the great zebrafish system to now bring together two genetic model systems and see if we can start to get more complicated in the biology that we're trying to describe, and then ultimately pull apart, use some of the tools possible. So how is it that Tetrahymena might be purveying viruses? So we can imagine cases where the viruses are degraded, as we saw with the phage and some of the other enveloped and non-enveloped viruses, that it's modified. That's sort of the hint or the breadcrumb for thinking about chum salmon reovirus. And we'll also, sort of an obvious idea is that this is also a great concentrator when we're in sparse aquatic environments, as these ciliates are grazing on the carcasses of dead animals and tissues, that there could be almost like watering holes on the Serengeti, where a lot of biological activity is coming together for the first time, an opportunity for viruses to concentrate and spread. And as I hinted at, we'd love to understand how these interactions actually impact infections in the host. Is this all the way to vectored scenarios, or are there sort of intermediate states here? And so we'll use some of the tools that Kier developed when he was a postdoc in my lab before he came here. Okay, so this is probably a familiar image to many of you. This is the transgenic zebrafish that Kier developed when he came to my lab using the tool toolkit that was provided to us by Kristen Kwan. And so Kier hooked up the ISG15 promoter to GFP. This is a fish treated with interferon or injected with interferon. It lights up like a zebrafish. And so we wanted to ask if we could use this tool to understand, and then the virus that he discovered when he saw spontaneously GFP positive fish swimming around in the aquariums at Utah. We wanted to see if ZFPV might be a model system. So now 3 models, Tetrahymena, the virus, and the zebrafish to bring together to understand how some of these dynamics might work. Key point here, ZFPV, the zebrafish picornavirus, 15th cousin, 3 times removed from poliovirus, does not infect Tetrahymena. So we don't think this is all the way on the right side of that continuum of vectored biology here, of virology, but that maybe there's something going on in the middle. OK, so flash forward a year or two, and we tried to style out Kier's great GFP reporters by giving them a red eye. So we put M-cherry on the crystalline protein of the eye, and this allows us to sort of set up, I think, some interesting experiments here. So the first hint that we had was if we took Tetrahymena, exposed it to that picornavirus, ZFPV, and then looked for a green signal, we could see a little. It's pretty subtle, but you can see a little bit more. So we've really turned up the M-cherry signal here. If you kind of squint at the green, you can see without Tetrahymena, the zebrafish with especially in the GI tract, you can see some of that GFP ISG15 reporting, but more so when you added Tetrahymena. So then Katie set up an experiment. It's one of my favorite experiments that we've done in the last few years. And it goes like this. So she takes some of those ISG15 fish, the original ones that Kier engineered, and infects them with ZFPV. She sorts out the GFP positive ones, and then she puts them into plates, a single green reporting ZFPV1 infected fish with a plate of red eyes. They're all cured of the virus, so they don't have it in there. And then the only difference in the 2 dishes, the experimental dishes, is the presence or absence of Tetrahymena, that ciliate that might facilitate or purvey the infection. Then 24 or 48 hours later, she grabs all of those red-eyed fish and asks them, are they starting to report an immune response? What does this mean for the sort of quality of the virus infections, of the ZFPV1 infections? So if we look at ZFPV1, we're now reading on the y-axis the relative copies of the virus. You can see even at 24 hours, there's a bit more virus being purveyed to the red-eyed fish, which also sort of persists at that 48-hour time point. I think even more interesting is what happens on the immune side. So at 24 hours, it's pretty even. We're looking at the Tetrahymena plus dishes in blue and the Tetrahymena minus in gray. But by 48 hours, you can really see a massive difference in the greenness of the fish and also the amount of ISG15. She's also done this now, and so here's the GFP data. About 2 times as many fish of the red-eyed fish report green. And the amount of the interferon-stimulated gene is really high. This is one. We've done 2 other interferon-stimulated genes, very much the same results. So much so that I'm not showing that data, just sort of phenocopies the ISG15. So 2 things are happening here. There's more virus that's being conveyed with Tetrahymena, in the presence of Tetrahymena, plus the immune response is turned on to a much higher level. And so this starts to get at this notion of the Tetrahymena doing more than one thing or adding a lot of complexity, not as sort of a traditional vector, but actually potentially stabilizing the infection by also stimulating an immune response at the same time that might bring the virus infection into a balance that might not otherwise exist without this third party in the operation. This is very different if we don't have Tetrahymena, but we upregulate interferon without Tetrahymena. So these are zebrafish, transgenic zebrafish that have a heat shock promoter driving interferon signaling. And so if you heat shock these fish, or even if you just let the sort of small amount of interferon that leaks through constitutively from the heat shock promoter, you can see that ZFPV is down and that the immune response is up. And so this is some data we have. Depending on if you heat shock these, the ZFPV can go away altogether. And so that's some of our emerging evidence that there's sort of more going on here in terms of the stability of the infection. OK, so let's go back to this idea of protists or ciliates or tetrahymena as purveyors of viruses. So we have, and this is sort of our working hypothesis for what's going on. Still very much a hypothesis. I'll show you a little bit of evidence, but we're still working kind of in this direction to see where the evidence takes us. So the notion is that actually some of these highly differentiated and specialized secretory pathways might be at play to help promote this sort of stabilized infection situation. We think the process of phagocytosis, this is the concentration of all of the particles, including viruses around the ciliates, is immunostimulatory. So as the viruses are processed or activated, as we saw with the salmon virus, chum salmon virus, that this is an opportunity for these things to be presented to the immune system as an uptick that way. At the same time, there's these other pathways out at the surface, at the cilia, that sort of organization of the cell I showed you from that Golgi marker. There's also incoming traffic here, clathrin-mediated endocytosis. And it might be the uptake of the viruses, not in that sort of proteolytic pathway, but in the secondary pathway that then somehow presents the viruses to the host when they're consumed, the zebrafish in this case, that actually promotes virus transmission or the virus infection. OK, so to start to get at this, we first took away phagocytosis. So if you treat tetrahymena with cytoclasmin B to inhibit actin dynamics and colchicine to inhibit some of the microtubule dynamics, you can block phagocytosis. And this is very specific. So endocytosis, or clathrin-mediated endocytosis, which is a dynamin-dependent process, I have to point that out for Sandy Schmidt in the audience, is independent of this pathway. So clathrin-mediated endocytosis is still running. So we can check, kind of a gut check if this is working by mock treating and then giving the ciliates India ink, which they concentrate into these phagosomes. Within minutes, you can see all of those black dots. Those are the phagocytic lysosomes in the cell. Treated with cytoclasmin B and colchicine, they don't get in. So then we very simply ask, OK, what happens to the immune responses when we add that zebrafish picornavirus to the menu here? And what you can see is that with phagocytosis blocked out at the far right here, that the ISG signaling is way down. OK, so next we wanted to ask, what if we just mess up the tetrahymena altogether? So we started to do freeze-thaw cycles in the same sort of experimental setup here. And so here the data, I would say, gets a little more murky, but I think also a little bit more interesting as well. So if we incubate the tetrahymena with ZFPV for 4 hours, it sort of mimics that first experiment I showed you. Wash them a bunch, and then freeze-thaw 3 times. The virus does OK. So you can see ZFPV across the 4 treatments here, tetrahymena plus or minus, freeze-thaw plus or minus, that the viral levels, as we're reading that out in the genome copies, is about the same. However, the ISG signal, or the immune response, again, is way down. And so this is evidence that there's more going on here. So in the processing of the virus depends on the vector, or I don't know the right words here. So we're still working out the language. I'm going with purveyor or conveyor. Anyone who has feedback on what we're seeing here, that would be great. Very welcome. But the tetrahymena has to be actively alive for the immune response, for these viruses to be processed in a way that is immunostimulatory. We've also done the obvious experiment. I'm not showing it. For some reason, I thought I didn't have as much time. But we've done UV cross-linking to inactivate the virus. So we're taking away the conveyor or the purveyor of the virus. If you mess with the virus by UV treating it, you see that the ISG, so certainly the virus level is way down. And maybe surprisingly, the immune response is also down as well. My sort of hunch would have been that the immune response would still be up because you could just chew up this sort of inactivated virus. But it looks like there's something more going on with the tetrahymena with the ciliates to process this or activate the immune function as well. OK, so the next obvious experiment, which we're building up to do, sort of future directions here, is to start messing with clathrin-mediated endocytosis. And to sort of formally test the idea that the virus might get into the cell that way and then is presented sort of in a Trojan horse kind of situation as the zebrafish are consuming the ciliates as prey. And so one of the things we can do is deploy a dominant negative version of clathrin to specifically block this route of endocytic traffic. So clathrin that forms those sort of classical cages around the clathrin mediated pits also has this great geometry across the surface of tetrahymena. I mean, these are sort of the poets of cell biology. And so you can watch this biology play out. In red here is centrin. So these are the basal bodies where each of the ciliary rows emerge, and you can see that juxtaposition of endocytosis there. So here the marker we're using is the fluorescent dye FM143, which has been classically used in mammalian systems to sketch out sort of the dynamics of endocytosis, and it works here as well. So if you express the clathrin hub, just a dominant negative fragment of the clathrin triskelion, you can see that cell next to the one where you have all that signal is one expressing the hub where it can't get into the cell. And so the obvious question is, what does that mean for the virus here as well? One obvious question that sort of might be coming to mind is, okay, you're sort of throwing together this soup of genetic model systems, but does this really happen out in the wild? And so very little is known about tetrahymena in South Asia and Southeast Asia, where zebrafish are endemic too. So this brings us to these freshwater environments to ask whether zebrafish is seeing, does the virus play a role here? And so one idea is that ZFPV, while it seems like this is something that Keir discovered just sort of from messing around with our fish in a facility in the US, could this be happening in the wild? And is it possible that the virus has evolved to manipulate some of this cell biology in order to dictate the ability of this thing to persist as stable infections in zebrafish around the world? And so we don't yet know. We're sort of thinking about mounting some trips to do some collecting, both of wild zebrafish and to ask what are the ciliates that are home sort of in these environments to see if this sort of purveying of virus or these relationships exist in the wild. I have one sort of a hint that this might be the case. This is a recent publication from Ming-Chao Yao's lab. He's one of the founders of molecular biology in tetrahymena and recently published some field studies collecting bladderworts that you see here. These are bladder traps from bladderworts, and inside of them, they're capturing prey. These are from irrigation ponds in Taipei, and they found 9 new species of tetrahymena. So basically, wherever you find, in aquatic environments, wherever you find zebrafish or other fish, you will find these ciliates that could be sort of conducting this biology. I also just really wanted to say bladderwort trap in a talk for the first time. Again, I'm in Keir Ball country, so I'm trying to really hit my sort of eccentric biological credentials here. Okay, so to start to zoom back out a little bit, the idea here is that there's probably, in addition to sort of the obvious kind of black and white sort of virus transmission chains, direct contact, certainly there's inactive sort of facilitation of virus infections, whether particles are traveling or hitching a ride on dust or other fomites. On the other end of the spectrum, we have these highly vectored scenarios, mosquitoes, where the virus can actually replicate in both hosts and promote the virus chain. A lot less, I think, in between on this continuum, and I think these aquatic systems, and the ciliates in particular, all of that diversity of life that we barely have scratched the surface of understanding could also be very much in the game here. What I do want to return to, I'll close on actually 2 ideas. So the first is that while the terrestrial sort of virology might seem at first to us very different than the aquatic virology, certainly in these 2 cases, there might be a link that's just sort of like right in front of our noses. And that is the aquatic stages of arthropods, right? So in addition to mosquitoes, you've got ticks, other insects that are involved in vectoring. A lot of these have nymph stages that are aquatic, which is the perfect opportunity for these vectors or these purveyors of viruses to actually see each other and for viruses to sample the air-water barrier. Metagenomics in the last decade or so has taught us that every major virus class that you see in our lungs, that you see the respiratory viruses of terrestrial creatures, are also out there in fish. Influenza viruses of fish, nidoviruses of fish, pox viruses of fish. The second sort of point that I want to close on is, okay, maybe zebrafish coronavirus is hitching a ride on ciliates like tetrahymena, but what is it that infects these ciliates in the same place? And of course, there's been a gold rush of interest in the giant viruses that you find in a lot of protozoan systems, and really reflecting, I think, again, the life histories of ciliates and other protists that are acting as sort of mixing zones for all of these biological particles. If you go into the genomes of these giant viruses, it's just sort of like an inspiring tour of cell biology from eukaryotes, bacteria, archaea, all of the above, and where the viruses are almost acquiring these genes and trying them out like playing cards. And so are there also viruses in tetrahymena? If that's the case, it hasn't been described yet, but if that's the case, again, we have potentially the advantage of a model genetic system to tease apart some of this virology. So there are some hints out there. This draws from some local work from about a decade ago, Alex Greninger and Joe DeRisi's lab. This was, I think, before the e-foiling days, but when they were sort of hanging out at the San Francisco sampling the sewer water. I think it's now, I asked ChatGPT-5 to draw a picture of Joe DeRisi on an e-foil with a butterfly net sampling water. What ChatGPT came up with is absolutely like obscene and ridiculous. I can't show it at a family event like this, but you can imagine the kind of virus sampling that's going on right in this neighborhood. Go back 10 years ago, and sorry, Joe, and Alex was generous enough to like find an old hard drive that had all kinds of sequence from these San Francisco sewer water samples. And in there are two proposed viruses, cilioviruses, that really caught our attention. RNA viruses, so these are not the sort of DNA virus supertankers, the giant viruses that are probably in the game here with ciliates as well. But there's some really interesting features of these viruses that really captured our imagination. So if you take the ciliovirus 2 polyprotein, for example, 300 or 3,000 amino acids predicted, and just run it with a standard genetic code to sort of predict the open reading frames, you're going to look at this and say, you guys don't know what the hell you're doing. This is just junk this is genetic junk that you've found here. However, if you play one trick, which is, so there's no open reading frames across the polyprotein. Ciliates, however, have their own genetic code. So 2 of those stop codons go away, and these are glutamines. So if you just decode it with the ciliate code, all of a sudden it flies right into focus here. This is clearly a virus of ciliates, and in fact, sort of guilt by association, there's all kinds of sequencing tetrahymena reads that are sitting here. So Katie has took the liberty of synthesizing this virus. We're trying to launch it off of RNA now. So far, it hasn't sort of taken our invitation to start replicating, but we think it is immunostimulatory, which offers a whole nother sort of avenue into figuring out what is the immune system of these ciliates. And I'm going to end with 2 ideas in that direction. One is that you don't necessarily need these sort of fancy hundreds of repertoires of interferon-stimulated genes. You can use some of your core cell biology, some of your core molecular biology. So in this case, in order for a virus to come online in a ciliate, it has to know the genetic code. And so this might be sort of from if you back out and think at sort of an evolutionary kingdom level, a constraint on virus evolution to move between a ciliate, for example, into a fish as something that's vectored like a mosquito vector, like an arbovirus. In order to pull off that trick for these viruses, they'd have to be code breakers. They'd have to be code switchers to pull it off. That also makes us think about this core biology. So the proteases that you see in the phagosomes of these, this is also, as we look at some of that early data about how the viruses go away, another maybe breadcrumb about what immunology looks like for these protozoans. Again, if this is true, then if we think about viruses and how they evolve around these corners, then you can imagine very quickly that some of these viruses will use the protease processing to process themselves to become more active. Evidence of that in the chum salmon virus. And I think when you look at these wild EMs of the giant viruses and all of the weird sort of features that are out on the surface of those cells, the stargates or whatever they are, you could just imagine those being proteolytically processed in ways that allow the giant viruses to gain access to the cytoplasm of the host cell to get out of that phagosome and to take over the ribosomes in the host. Okay, so I'm going to end there on the varied routes of infection, sort of glimpsing at these sort of systems that I think starts to use the reductionist approach or the logic of genetic model systems, but to offer a bridge to the ecology of these viruses out into the environments. And then finally, I want to acknowledge the Elde lab, other members in it. There's Katie in the lower right corner, some of these great scientific supporters along the way. I also want to close by acknowledging the headwinds that I think we're all feeling right now, the dissonance of trying to practice science, especially if you're at sort of the beginning of your career. What I would say is, things are, I heard someone say the other day, things are bad when, liberals start to quote poetry and things like that in their talks. I think it's even worse when you draw on Mr. Rogers, but here we go. When I was a boy and I would see scary things in the news, my mother would say to me, look for the helpers. You'll always find people who are helping. And I think that's really important in this moment as we sort of, in events like this, where we sort of build a new grassroots way of doing science together that draws on some of this historic, classic knowledge. Finally, I want to say that I think the stakes are high, but the stakes are worth it. So here's my kiddo trying out her hand at the laboratory whiteboard. I'm also using this as a transition to just apologize that I'm going to science and run here in an hour or two to hop on a plane to get back to Salt Lake City to get this kid into kindergarten tomorrow morning. Okay, thanks everyone. Happy to take questions.